Hi, this is Scott Oneto, and I'm a farm advisor with the University of California Cooperative Extension. And today we're going to be talking botany for gardeners. And this class is brought to you by the University of California Agriculture and Natural Resources Master Gardener Program. The goal of today is really to talk about how plants grow. And throughout the slides that I'll be sharing, I'll try to keep making an emphasis on how it really impacts plant growth and development. It's really just a fascinating area of study when we talk about plants, especially from starting with a seed and that seed germinating and then growing into whatever the plant is, and then potentially making a beautiful flower and then a fruit, and then inside that fruit, of course, would be a seed to start the next generation. So hopefully you'll find this course full of information. And if you have any questions at the end of the presentation, I'll have my contact information, and you can feel free to reach out either to myself or to your local cooperative extension office. So just a little bit of definition. When I think about what are plants, these three things come to mind. First, I think of plants as living organisms that lack mobility. So, as a master gardener, I think this is an important part to keep in mind when we're recommending plants for our clientele. So, choosing the right plant for the right place is really critical because, unlike other living organisms that if the temperature outside is cold, they don't have the option to go somewhere where it's warmer. They don't have the option to go in and put on a sweater or a jacket, or to shed clothes to stay cool. They really have to adapt to their particular climate. And so, we'll talk about different plants and how they are able to modify their environment to make it more suitable, whether it's losing water out of their leaves to cool the plant. Sometimes plants will actually have different colors so that they reflect more light. So they keep cool. So there's lots of modifications plants can have to make them more suitable for certain environments. The second thing that is really interesting about plants is that they have the ability to manufacture their own food. And so we'll talk about that in the next couple of slides. And then lastly, which is just a really fascinating phenomenon about plants, is that they have this remarkable ability to regenerate lost organs. And we'll talk in depth about this process and how it happens and what things influence the plant's ability to do this. So I mentioned that plants have the ability to manufacture their own food, and we classify plants as what we call autotrophs. So if you break down the term autotroph, autos is Latin for self, and trophos is Latin for feeder. And so these are the self-feeding plants. And plants have this ability to capture sunlight energy, and they take that sunlight energy often into their leaves, and then taking up water from the soil and then capturing carbon dioxide from the atmosphere, they're able to undergo a chemical reaction called photosynthesis. And that reaction results in the formation of a sugar. And that sugar is then used for driving all sorts of different cellular processes in the plant for growth and development. So whether it's the replication of cells, so those cells will ultimately make to new plant parts like leaves and to flowers and to stems and to roots. So basically the whole structure and the addition of new plant structures is really dependent on this ability of plants to make their own sugar. And then, as you can see in this little depiction here, a byproduct of that reaction is oxygen. So here we have our plant, and as I mentioned, it gets water taken up from the soil, and then it's taking in carbon dioxide through its leaves out of the atmosphere, and then sunlight energy is coming through, beaming through those leaves, and the leaves are able to capture that energy and then undergo that chemical reaction. So super cool, fascinating reaction. So, we'll start off talking about what plants are made up of. And, I'm sure many of us have all had some type of either biology or cellular biology class where we've talked about, the building blocks of life, which are cells. And so plants are no different. They are also made up of cells. And throughout the plant, it's just made, all of these different cells are like building blocks. And as we'll go through the next few slides, I'll show you some different types of plant parts and all of the different cells that are contained within those plant parts to make up the plant. So this was just a short little video that kind of shows, as we kind of zoom on into a root in this case, you can see all of these different cells that are underneath a microscope. So we're just looking at really, really close of the root, and you can see all these little building blocks. And so all of these are different cells, and these cells, of course, all have a different function for the plant. So they're all doing something different, whether it's, protecting the outside of the root, so to keep out, fungi and bacteria and other organisms from getting in, or perhaps it's a storage cell inside the root that's storing excess carbohydrates or excess sugars for the plant. So lots and lots of different types of cells, and those cells often have very specialized functions for the plant. So, this is just a diagram of what a cell looks like or could look like in a plant. And, as I mentioned, they're often very specialized and do certain functions for the plant. And so you'll see different types of what we call organelles, or these specialized structures. This is one here that I'm just highlighting over here. This is one called a chloroplast, and that is where photosynthesis occurs inside the plants. And so, often you'll see a lot of these chloroplasts in the cells that are, of course, in leaves. And so you wouldn't necessarily find, these chloroplasts down in the roots because, they're not exposed to sunlight, and so they're not undergoing photosynthesis in the roots. But in the leaves, of course, there would be lots and lots of these chloroplasts. So when we look at how these cells are organized, it's highly specialized, as I mentioned, highly sophisticated in the sense of, we have groupings of cells that form what we call tissues. And so it doesn't make a difference what kind of plant part we might be looking at, whether it's a root or if it's a stem or it's a fruit or a leaf. We'll see these groupings of cells that are of the same type, and they're doing the same function. And these groupings, again, of cells are called tissues. And then when we group several tissues together, we have organs. And so in this case, this is just a depiction of a leaf. And so from the top of this diagram down, we basically have from top to bottom of a leaf, and it shows some of the different organizational layers of that leaf. It's kind of like a cake, and all of the different layers of that cake. And so, if we start at the top of this leaf here, this is what is called the dermal tissue. Sometimes you may have heard of this as the epidermis, but basically these cells are all of the same type, and their main function, of course, is to provide protection to the leaf. So again, to not allow bacteria, viruses, any sort of bad things into the leaf. And so, we have the dermal tissue. We go down into this next one. You can see this one's identified as the ground tissue. And this one, of course, is full of chloroplasts to undergo photosynthesis. That's all these big cells. And you see they're really big, and they've got lots of space in there to store air, carbon dioxide, to undergo photosynthesis. And then if we make our way down even further, you see There's another, this is another tissue type. And then you have this one here that goes right down the middle of the leaf that almost looks like the trunk of a tree. And this is called the vascular tissue. And this is one that we're going to spend a little bit more time on discussing because it's just a really fascinating tissue type. And it's really important for how plants grow and develop. And so we'll talk more about the vascular tissue as we go through the next few slides. So here is some more information about that vascular tissue that I mentioned. So again, remember the vascular tissue is just a group of specialized cells. So it's cells of similar type that are organized to form basically this tissue type. That this tissue is then doing some sort of function for the plant. In this case, the vascular tissue is comprised of two main cell types. First, we have the xylem, which is a type of cell that conducts water. So it's responsible for moving the vast quantities of water throughout the plant. And then we have another one called the phloem, and that is responsible. So the phloem is another cell type, and it is responsible for moving those carbohydrates, those sugars throughout the plant. So those sugars that are made in the leaves through photosynthesis are then moved throughout the plant for growth and development through the phloem. This cross section here, this photo, this is, of course, you're probably all familiar with this. This is celery. The stalk here, the celery stalk, if you look at it in cross section, so if we just cut it in half and look at it, you can see these very distinct structures in the stalk, and these are the vascular tissues. So I'm going to talk a little bit about these two different cell types just a little bit more. I mentioned the xylem is one of the parts of the vascular tissue, and it's responsible for the movement of water from the roots up into the stems and the tissue, the stems and the shoots, and then out to the leaves. And the movement of this water is, of course, just one direction. This water moves upwards in the plant. So it moves from the soil into the roots and then up the plant. And I'm showing a couple of different diagrams here, and these are just some of the different types of xylem cells that you would see in plants. And I often think of xylem as plumbing, right? So as gardeners, many of us, of course, do lots and lots of irrigation work in our gardens, putting together either PVC or either poly pipe for running irrigation lines. And I like to think of the xylem cells as plumbing parts. And so, when we look at the xylem, there's a couple of different types of plumbing that we will see in plants. There's these ones here, which are vat plumbing. So I would kind of refer to these as like 3-inch PVC pipes. So big old fat pipes that can move lots and lots of water. And then we have narrower pipes. These here are more like 3/4-inch PVC pipes. So smaller pipes for moving less water. And how these work is basically these cells stack one on top of each other. Just like you would put PVC pipe together and use a coupler and attach them and make really long lengths of pipe, these xylem cells are basically situated the same way in a plant. So from the roots all the way up into the stems and out to the leaves, you have just these cells stacked one on top of each other, making this long continuous pipe going up the plant. The other thing that I'll mention, and I'll show you some pictures of it a little bit later on today, is that xylem cells are extremely important for the structural integrity of the plant, especially when they die. So the plant's continually making new cells, and some of these cells that are made become new xylem cells, and some become new phloem cells. And it's these xylem cells that as new ones are being made and others die off, they don't go anywhere. They stay inside the plant, but the dead ones basically become the structural integrity of the plant. And so I'll show you some pictures of that just a little bit later. So now let's move on to the other part of the vascular tissue. This is the phloem. And so the phloem, as I mentioned, is responsible for moving carbohydrates in the plant. So we have all of those sugars that are being made in the leaves. And once those sugars are made, they then have to get moved all across the plant to either be stored or to be used for some sort of growth and development. So it could be just in the replication of new cells, if the plant's just undergoing new cells and making new plant parts. So it's making new stems and making new leaves and making flowers. All of that takes energy and all of that takes carbohydrates. And so sugars are often being moved to areas of new growth. And then also, of course, they can be stored either in a storage organ like a root, or sometimes just extra storage cells in the stems for later use. And so this is a very bidirectional movement. So sugars are, moving laterally, they're moving up and they're moving down, and they all do it through a what is called a source-sink interaction. So the source, of course, is often the place where it's produced. So in many cases, it's the leaves that are the source because they're making tons and tons of sugars. And then the sink is that demand, that pool for carbohydrates. And so the sink is often those areas where the plant needs to use those sugars. And then the plant's able to draw on those sugars based on this pool or this need for sugars by the source-sink interaction. So this vascular tissue, this arrangement of xylem and phloem in the plant is arranged in a couple of different ways. And depending on the type of plant that we're looking at, you'll often see different patterns. And so in your readings of the Introduction to Horticulture or Botany for Gardeners chapter for this Master Gardener program, you'll read about these different classifications of plants, specifically the dicotyledonous plants, which we often call the broadleafs, and the monocotyledonous plants called the grasses. And these two groupings of plants are very different. The broadleafs have a vascular tissue arrangement that looks very, very different than that of grasses. And it's really important that we look a little bit into this because especially when we talk about broadleaf plants or the dicots, it's their arrangement of this vascular tissue that plays a critical role in terms of how plants actually grow. So we're going to spend a little bit of time kind of digging into that a little bit deeper and showing you how that arrangement of the vascular tissue impacts plant growth. So let's look at vascular tissue in a dicot or in a broadleaf plant. And so this, of course, is a carrot. Everybody's familiar with a carrot. And of course, we know the roots from the shoots or above ground portions of the plant. And so I kind of have a dotted line here right across from where the roots meet the shoots. And off to the left here is a picture of what the cellular structure or the cellular breakdown would look like in this plant. And so if we look at the roots here, if we take a cross section of a carrot, so if we just cut it in half right here, just go right across and look at it, this is what we would see. We would see the outside of the carrot right around here, right? This is the outside of the carrot. And we have all of these cells that basically all look the same all in here. These are all just large storage cells. So they're storing carbohydrates. And then if you look at the very center of the carrot, we have this very different appearing group of cells. And so this is the vascular tissue in a dicot, and all dicots look like this. So it doesn't matter if it's carrot or if it's an oak tree or if it's a rose bush, any broadleaf plant or any dicot is going to have a vascular tissue arrangement that looks like this, where in the center of the root, you have this grouping of cells, the xylem and the phloem, that's conducting water up the plant and moving sugars up and down throughout the plant. Now, if you kind of think of this, this bundle of xylem and phloem going right up the center of this carrot, and when it gets to the soil surface here, right at the soil surface, this bundle basically branches. So if you think of this original bundle kind of like as your forearm, and when you get to the soil surface and you get to the shoots, basically that bundle branches. And so now instead of having a single bundle that is right up the center of the stem, now we have these sort of fingers, these clusters of bundles that go around the perimeter of the stem. So this is the stem up here. And of course, this is the outer perimeter of the stem. And so just in from the perimeter of the stem, you have these very easily distinguished bundles. And all of these are xylem and phloem. So we have little clusters or bundles of vascular tissue that go all the way around the perimeter of the stem. And this is the same for any sort of dicot. Again, any broadleaf plant, the stem is going to look like this. So we'll look a little bit more about what's actually in there and what they're doing here in a second. But I just wanted to show you what that looks like from the whole plant level. So now let's get into where the xylem and phloem is in that. So here's just a little bit more close-up view of that carrot root. So again, the outer perimeter of the root out here, and then in the middle, of course, is that vascular tissue. And the xylem is the, you can see there's kind of an X here. And so the X is made up of xylem cells. So again, these are just tubes that are carrying water all the way up the plant. And then in the apex of this X, so all of these little small circles here, these are all very small tubes of the phloem. So these are the carbohydrate or the food conducting tissue. So again, just think of all of these as continuous pipes going up the plant, but all these groups of pipes. So we have a group of pipes that are in this X formation carrying water up the plant. And then we have these little clusters of pipes. We have basically one big bunch of clusters there, one cluster there, one cluster there, and one cluster there. And those are the phloem pipes moving, again, carbohydrates around. So if you look at a carrot here, if we did a cross section of a carrot, and then we were to just take a thin slice here, and hopefully you can see, I'll just kind of point out there really, really nicely in the center here, you can see that vascular tissue. And I'll just draw a little ring around it. So there is that vascular tissue right there in the carrot. And then we have in there, we have this X, which makes up the xylem. And then we have the phloem in the apex of all of those. So again, all of the dicots will look like that. Now, if we go into the above ground portion here, again, just a close-up of what I've already showed you. We have these clusters, or what we call bundles, going around the perimeter of the stem. So again, this is the stem of our broadleaf. And within each one of these bundles, again, we have xylem, and the xylem is towards the inside of these bundles. So towards the inside of the stem, all of these white circles are pipes of xylem cells. And then just above or towards the outside of that stem, we have phloem cells. So these are all phloem pipes. So you can see these lots and lots of little clusters with numerous bundles or pipes of both xylem and phloem in the perimeter of the dicot stem. And now when we look at the other big classification of plants, I mentioned there's these monocots or the monocotyledonous plants, which we often think of as grasses. Their vascular tissue arrangement looks very different. So I have a cross section of a root here on the left and a cross section of a stem here on the right. And I'm not going to go into any more detail on the monocots. Just wanted to show you that they are in fact different. So In the root here, the large round ones here is the xylem, and then we have the phloem's cells in between. And so it makes this sort of concentric ring around the root of the grass. And then when we get up to the stem, each of those kind of branch off, kind of similar to what I described before, but more of I guess you could say, randomized pattern for the stem of the grass monocots. And so again, I'm not going to spend any more time describing this one because it's really the dicots that gets kind of interesting with its arrangement and how that arrangement of the vascular tissue is important or plays an important role for several broadleaf plants in terms of growth. So we'll continue on with talking more about the broadleafs here in a second. So I want to spend just a few minutes talking about how water enters the plant, both water and minerals. We're getting, of course, lots of nutrients being taken up by the plant that come into the root. And so let's talk a little bit about how that happens. And so this particular picture is just a close-up of a root, and doesn't make a difference, what plant it is, but it's pretty much showing a picture of a dicot. So we have, this is that center vascular tissue here at the middle of the root, and this is, of course, the outer perimeter. And so these little appendages out here, these are what we call root hairs. So of course, you probably have all seen root hairs, especially on a carrot. They're those little white, whispery type whiskers that stick out off of the carrot. Those are just individual cells that are helping with the overall surface area of the root to be able to take in more water and minerals from the soil. And so if we look about how water is actually taken up into the plant, it can actually happen a couple of different ways. And so the first way is a process, basically what is called osmosis. You've probably heard of that term before. And it's a process in which water will move across the cell membrane or the cell wall into a root cell, and then it'll move across to the next cell adjacent to it in that continuous pattern all the way until it gets to the vascular tissue. And so it's so you can kind of envision here, water molecules out here, they get taken in by this cell, and then they get basically transferred across cell walls from this cell to this cell, from this cell to this cell, from this cell to this cell, until it reaches the xylem. In this other way, we have more of a diffusion process where water is moving between cell walls. And so you can kind of see it shown here where we have water molecules moving kind of in between cell walls here, in between all the way again to the xylem. And so the thing that I want to point out with that is this is a very slow process, but it's once that water gets to the xylem, and you think of again, how I described the xylem, where you have these basically cells stacked on top of each other to make this continuous pipe from the roots up into the shoots. Once it gets into there, it's an extremely fast and efficient way of transporting water. Whereas it may take minutes for a water molecule to move across just this little short section of a root from the soil to the xylem. Once it gets into the xylem in the roots, it might be able to move hundreds of feet up a tall tree within seconds. So hopefully that kind of gives you a visual of just how quick. And you can kind of think of it, if anybody's ever used a garden hose or any other type of hose to siphon anything. If you've ever had to move water and you've used the basically suction, and you suck on the hose that's in a bucket, and then you move that water to somewhere else, or that fluid to somewhere else. That process is very similar to what we're trying to envision what's going on in a plant through the xylem. So we have just very, very small pipes that are going up the plant that's drawing this water from one reservoir of of water that's in the soil up into the top of the plants. So let's show this little video that kind of describes that process.
Plants transport water and nutrients from their roots to their leaves through a vascular tissue called xylem. These are dead, empty cells which connect end to end, forming long continuous tubes. Because water molecules carry a slight electric charge, they're attracted to each other. This natural attraction, known as cohesion, links the molecules in long continuous chains. At the leaves, water escapes the xylem and evaporates through microscopic pores on the surface called stomata. As each molecule breaks loose, it pulls up another molecule to take its place. This continuous pulling tension extends all the way down the xylem tube to the roots. Here, the rising chain of molecules keeps pressure low, allowing water and nutrients to enter through osmosis.
So I think that's a great video that just kind of shows some of the properties of water that we're talking about in terms of how we're able to, again, visualize how water can actually moving against gravity up to, often hundreds of feet tall in some of the tallest trees. And so the video mentioned a couple of different things. One, the idea of cohesion, which is water molecules have a tendency to want to stick together. And so if you pull on one water molecule, it'll pull on the next one, which is attached to the next one, which is attached to the next one. So we have this long chain of water molecules. So as plants are using water, whether it be through photosynthesis or in the transpiration of water out of the leaves to help keep it cool, that using of water molecules is again dragging or pulling water up these columns of xylem tubes. And then there's also another property of water called adhesion, which also is playing just as a critical role in this process. So where cohesion is the joining of water molecules, adhesion is the attractiveness of water molecules to other surfaces. So, like if anybody's ever put a straw into a glass of water or any sort of liquid, you'll often notice that the water inside the straw often climbs up the straw a little bit higher than the surface or the level of the liquid in the glass. And so that attractiveness of those water molecules, not only to themselves, but also adhering to the sides of that straw, play a critical role in this idea of water moving again against gravity. So there's no pump moving water up the plant. It's just these forces within the plant that are driving this process. And there's actually a very interesting mathematical equation that describes, or that you can actually calculate this this gradient of water movement in plants, and it's called the water potential gradient. I've got a picture of it here, and it's showing that this movement of water is occurring against this gradient, moving from what we call a high water potential gradient. So we have, basically you can think of it as we have lots of soil moisture, so we have more water in the soil than water that's in the atmosphere. And so there's this difference, or this gradient between those two. And so as we lose water out of the leaves, we pull water against this gradient from the water that's down into the soil. And so in agriculture and also horticulture, we can often use this water potential gradient for measuring either how drought tolerant a plant is, or if a plant is in need of an irrigation cycle. And so there's equipment that can actually be used to measure what the water potential gradient is, and determine whether or not a particular plant might need an irrigation. So this is a fun little video clip showing how a plant can revive itself when it becomes drought stressed, almost to the point of what we call the wilting point in plants. And so when water is taken up by a cell through osmosis, the plant cell basically becomes very filled with water, pushing against the cell wall, and we call that turgor. And there's basically a turgor pressure. And so, basically it's what causes the cell to expand to a point where it's, firm. And as a plant loses water, it becomes more drought stressed and it loses that that pressure. And so that's why plants have a tendency to wilt, because they just don't have the water in the cells to keep them rigid. And so if you think about, again, inside these plant stems here, we have lots and lots of little tubes, the xylem tubes, that are carrying water up the plant. And if you think about the water molecules that are inside those pipes, they're basically attracted to one another, right? And so as water molecules are leaving the leaves, it's pulling on the water molecule next to it, which is pulling on the water molecule next to that. And there is this tension. So again, this water potential gradient. So as we're pulling the water up these tubes, if that tension becomes too great, you can imagine what will happen is pop, they snap. Those water molecules are just under so much tension that they eventually just snap. And if you get too many of those to snap, that is when you get to the point where a plant reaches its wilting point. Because even if you then pour a bunch of water onto the soil, if you've lost all of those pipe connections that's pulling that water up, you can't always get those water columns to reconnect. And so that's when you reach the wilting point of a plant, is where it's gone to the point where those water columns within the xylem have all snapped and it's not able to regain that turgor pressure. So let's talk now a little bit more at a larger plant scale about plant growth, and where actually does plant growth occur? Is it occurring at certain regions in the plant, or is it just occurring everywhere, right? We've got this big old tree here and it's growing, right? It's growing and it's making new leaves and it's flowering and it's making, new branches, and it potentially is making fruits. So all of this is happening continuously. And so let's talk about where that plant growth is actually occurring. And I'd also like to just, think about this idea of, if we were to drive a nail into the trunk of the tree, say we put it in at 4 feet off of the ground, and we were to come back in 20 years, where is that nail going to be? Is it going to be at 4 feet, or is it going to be at 6 feet or 8 feet? A tree is growing over time, right? So, you think about a tree and it's just getting bigger and bigger and bigger and bigger. So where would that nail be? So if we dig a little bit deeper into this idea of plant growth, and what I mean by plant growth is I am talking about the addition of new plant parts. So the plant is making new parts, whether it be making new leaves, making new stems, making new roots, making flowers, making fruits. All of those things involve the replication of cells, and that cell division is called mitosis. So mitosis is basically, you have a cell and it replicates and it makes additional cells. And so we have a couple of places where cell division or mitosis occurs, and they occur at these regions called meristems. And in plants, we have 2 main meristems that we're going to talk about. The first is what is called an apical meristem. And so these apical meristems are responsible for cell elongation and cell division. And so this is where we get the formation of all the plant parts. So whether it's new stems, new leaves, roots, flower initiation, whatever the plant part is, these occur at the apical meristems. And it's important to note that these apical meristems are found in all plants. The location may vary a little bit, but they are found in all plants. And so if we think about a dicot, so again, remember these dicots, these are those broadleaf plants that I talked about earlier. So dicots have apical meristems that are found at several locations. We're going to start at the top of this plant here. And at the top of the plant, we have a apical meristem. So at the top is our shoot apex. This is our shoot apical meristem. Some people call it the terminal bud, but that bud that we often think of is a apical meristem. And just like the top of the plant at the bottom of the root, so if it has a long tap root, at that tip of that root, it's going to be a apical meristem. And then going back to the top of the plant, all of those lateral buds are also apical meristems. So the lateral buds along the plant shoots and branches, along the stems, those are all apical meristems. And then also all of the root tips have apical meristems. And again, their main function is to undergo mitosis, so undergo cell division. And as it undergoes cell division, it's making new cells. Those cells differentiate, or they become some sort of cell, whether it's, as I mentioned earlier, it could be an epidermal cell. It could be a cell that's in the ground tissue. It could be a vascular tissue cell. Doesn't make a difference, but that all happens in these apical meristems. So if we think about the other main grouping of plants, so I mentioned where in broadleaf plants or the dicots where those apical meristems are placed. In grasses or monocots, it's a little bit different. It's just they occur in the above ground portion. It's down here at the base of the plant. So, right where the soil interfaces, you have your little apical meristem kind of tucked under there. And that makes sense, right? If anybody's ever had to mow a lawn, you have these blades of grass, right, that are growing and they eventually get tall and you have to cut them. So we come in and we mow the grass. Once we mow the grass, those blades that you just cut, they're not going to ever grow any taller. You cut them at, say, 2 inches, they're going to stay at 2 inches. But the new growth that eventually outgrows that grass that you cut, or those blades that you cut, they're all originating from down here at the apical meristem. So new cells are going to divide. Those cells are going to elongate, and as they elongate, they push up and pass the blades that you already cut, and then you have new grass that you then have to come in and mow, and then that cycle just keeps on continuing. And then likewise, all of the root tips have also apical meristems. So this is a super cool plant, right? Many of you are probably familiar with Brussels sprouts, and if you've grown Brussels sprouts, you've been able to go and harvest all of the Brussels off of the plant. And so if we think about what these are apical meristems, right? These are lateral buds. They're just massive lateral buds that occur on the plant. And inside these are apical meristems that are undergoing mitosis to make new plant parts. So if we are to look inside an apical meristem, this is what we would be looking at. So I'll show you inside of a Brussels sprout here in a second, but it's going to look very much just like this. This is the inside of an apical meristem under a microscope. And what we see here, that we have right in the center here, we have this dome. And this dome is basically the location where mitosis is occurring. So this is where cell division is occurring. And so as new cells are being made, so new cells are being made right here, they over time elongate and get a little bit bigger. And as they elongate, they push this dome up, and more cells get made, they elongate, they push this dome up. And so if we go down, these are older cells. So these cells were made before these cells were. And so likewise, the stuff all attached down here is also older. So this particular thing sticking off here on your right, this is a leaf that was made at this dome when the dome was down here further. And then just only thing that has happened is time. And so as time occurs, the cells elongate, they start to get filled with sugars and they expand and get longer and bigger. And as they do that, you start seeing the shape. So this is an older leaf, and then we have some stem tissue, and then there's another leaf, and then of course it makes a bud here where there's a leaf, then you go more stem tissue, and then here's another formation. This is just the beginning of what a leaf is. So all of the cells that are in this leaf right here are the same number approximately of cells that are in this leaf. These are just older, so they've had a chance to elongate or get bigger. So that's all happening in these apical meristems. It's making new leaves, it's making new stem tissue, it's making a new xylem and new phloem and new buds. All of that's happening right here. And so we can see that, of course, in a Brussels sprout. So we've got a cool little Brussels sprout right here, right? And if I just cut this Brussels sprout in half, so right here is the dome where mitosis would be occurring. And then if you make your way down the dome, you can start seeing larger leaves that were formed. And so all of this is basically, you have a whole plant that's just kind of tucked away in this little bud that's just waiting for it to eventually pop open and continue to undergo mitosis and continue growing. And so when you think about dormant buds, that's all what's packed inside that little bud. And so if we go back here and look at the roots, the same sort of things happening. So this is just the tip of a carrot root and just tucked in back from the root tip. So we have the root tip down here, and then just up from the root tip, basically where this color transition occurs is, of course, the root apical meristem. And so again, that is where cell division is taking place. Before we move on to the next one, I'd like to show you one more example here. This is a Photinia. So this is a Photinia shrub here. And what we're looking at, of course, is a bunch of new growth. So if we look here towards the tip, so right in, peel this back here. So right here is, of course, tucked in here is the shoot apical meristem. And as it undergoes mitosis and makes new cells, the tissue or the stuff that's making inside that will begin to expand. So these leaves are getting longer, they're just getting older, so they're expanding, and they start falling out of that bud as it just keeps on making new cells, and it pushes that dome up. And as it keeps on making new cells and pushing that dome up, more leaves and more parts start falling out. So these are some new leaves that are just falling out. Here's some slightly older leaves that are they're still expanding and getting longer. And you keep on going down the stem. And again, you have more stem tissue that was all made, more leaves. There's little buds attached to the stem, more leaves, more stem. And you can see that thing came all the way back down from here. And then, of course, all of these leaves were made out of that same bud when the bud was, of course, way down there. And then, likewise, you can see, so where a leaf attaches to the stem, I'm going to zoom out here just a little bit. So you can see this old leaf here attaches to the stem here, and there would have been a little bud right there. And at some point in the last few weeks or the last month, this bud started to grow, and it kept on growing, and as it grew, it made stem tissue, and of course it's making leaves, and that little bud is just tucked away right in there. So that bud right there at some point about a month ago was right down here. And the only thing that's happened is mitosis and making more cells, making more plant tissue. And as it makes new cells, those cells expand, and it just pushes that little dome or that little bud continually up. So now let's move on to another type of meristem, and this is one that we're going to call the lateral meristem. So we talked about how plants get taller, put on new stems, make new leaves, make new roots, all of the different parts within a plant. Now we have to talk about how some plants, and I said the word some because not all plants can do this, but how some plants become woody. And so plants become woody through this process of what we call a lateral meristem. And these lateral meristems, of course, are only found in woody plants. So plants that can get woody can form lateral meristems. And these lateral meristems are responsible for the increase in girth, so making plants bigger, thicker. So if you think of a tree, the trunk getting bigger, those branches getting bigger. And if you take this just a step further, so not all plants can become woody, right? There's just a certain type of plants that can become woody, and those are dicots. So only dicots can get woody, but not all dicots get woody. We can have annual dicots, right? So anything from, I showed you a carrot. A carrot is a dicot. It doesn't get woody, but if you think of shrubs and trees, those are all dicots, and of course they can get woody. So if we go back to several slides ago, I was talking about the vascular tissue, and I was showing you the orientation of the vascular tissue specifically in dicots. So if you remember in a dicot stem, remember the orientation of the vascular tissue and dicots all looks the same, right? We have these bundles, these little bundles of vascular tissue that make their way around the perimeter of the stem. And so here is just a different diagram, different than the one I showed you before, but each of these little bundles here that represent this red color and this blue color, these are vascular tissue. And we have xylem, which is the red. The red are the xylem cells, and the blue is the phloem cells, or the tubes, remember? And they're just clusters of tubes that kind of go around the perimeter of the stem. So when a plant, when a particular dicot plant is ready to become woody, there's something that occurs in between the vascular tissue of these bundles. And what happens is the cells in between the vascular tissue becomes meristematic. And if you remember what meristematic means, that means that cells can undergo mitosis or cell division. When a plant is ready to start becoming woody, these cells start undergoing mitosis, and they start making cells to the left and to the right. And if this one makes cells to the left and right, and this one makes cells to the left and the right, eventually what happens is you form this meristematic ring. You get this ring of tissue, of cells, that becomes the lateral meristem. And once this lateral meristem is formed, it then starts undergoing cells to the inside and to the outside, right? So this ring formed, and now it's going to start undergoing mitosis and make cells to the inside of this ring and cells to the outside of this ring. And so over time, it'll look like this. So this ring that formed is making cells, which are xylem cells, to the inside. This is all this red tissue, and it's making new phloem, or food conducting tissue, to the outside. That's all this blue. And as this ring continues to undergo mitosis, making new cells to the inside, new cells to the outside, as those cells expand and elongate, it continues to push this ring out further and further and further, which ultimately is increasing the thickness or girth of that plant. So this is just a picture of a small little branch, and you can see right between the color change, the drastic change in color between the red and this tan, that is the lateral meristem. And so all of this tan tissue is xylem, water conducting tissue, and all of this red tissue is phloem, food conducting tissue. So let's just take a little bit closer look at that. So I showed you the vascular tissue, the lateral meristem here, where it's that distinction between the red and the tan tissue. And if we look at it just a bit closer, you can see the cellular structure, right? You could start seeing the actual xylem tubes, and you could start seeing the phloem tubes. And the thing I would like to point out again is, remember early on when I described the xylem cells, I said that, xylem cells don't live forever. The plant's continually making new xylem, and it's the cells that are right adjacent, really close to this lateral meristem. Those are the ones that are actively transporting. So it's these cells, these xylem cells right here, all along this ring, that are transporting water. It's these phloem cells right along the ring that are transporting sugars. And then if we look at some of these older xylem cells, these are all dead xylem cells all down in here, but these are the structural support of the plant. As these cells die, they become super rigid, and they are the ones that are providing that heartwood of the tree. That's that woody part of the tree. That's all dead xylem cells. And the other thing I'd like to point out is that you'll also notice that you can see very clearly the different sizes of those pipes here in the xylem. So as I showed earlier on, we have basically two different pipe sizes when it comes to xylem cells. We've got the big fat pipes that move lots and lots of water. We've got the little skinny pipes that move a lot less water. And if you look at the pattern of those big pipes versus little pipes, you start seeing a sort of a repeating pattern over time. And so when this vascular tissue, or this lateral meristem, was down here, right? So if we go back in time and it was down here, it was making all these big fat cells. And so if you think about, timing of year and what plants are doing, when is there lots and lots of water in the soil? When's there a big demand for moving lots of water in a plant? Well, it's typically in the spring. So, it's warm, plants are undergoing super rapid growth, making lots and lots of new leaves and lots of new stems. And so there's lots and lots of water in the soil. So the demand for water up of the plant is super high in the spring. So this is usually springtime when plants are making lots and lots of those big xylem cells. And then as we start moving into summer, growth begins to slow a little bit. Water demand, water in the soil becomes a little more scarce. And so we start seeing less big pipes and more little pipes. And as we get into, later in summer, again, less growth, less demand, less water. So we start seeing even fewer big pipes, more little pipes. And we get into fall and into winter, some plants will become deciduous or are deciduous. They drop their leaves. They kind of just shut down all completely. The plant's still growing, but the demand for water is much less. And so we see very little big pipes, and it's mostly little pipes. And then the cycle starts over again, right? And it's that repeating pattern of, big pipes to little pipes, big pipes to little pipes that we can very clearly see in woody plants. And we often count the rings of a tree to count the number of years of growth. And it's that repeating patterns of big pipes and little pipes over time that makes it very easy to count the age of a particular woody plant. So just going back to this particular slide, we talked about, where plant growth is happening. It's happening at these specific areas called meristems. So, of course, we have our apical meristems, which, are at the apex of the, terminal leader here, but also at the branch tips of all of these branches. And then all down these branches, we have lateral buds that are also apical meristems. And then down in the roots, we have all of these apical meristems. So the elongation of the plant is happening at all of those apical meristems. And then the increase in girth is happening at these lateral meristems. And so I'm going to go back here quickly to the Photinia bush that I have, because I'd like to show you just how that transition happens. And so it's very fluid, right? And so, up here, I showed you this new growth that was occurring. So this was all new growth, and the new growth has a tendency to be very flexible, right? So this is all flexible new growth. And so you can envision what the vascular tissue looks like in this new growth. It's, if we take a cross section of this growth, you have those little bundles of vascular tissue that kind of forms around a ring around the perimeter of the stem. But as we go down this stem, all of a sudden we get into this thicker, more firm growth. And so if I try to bend, of course, this stem, right, it's much, much thicker and firmer. And so this is where we have the transformation of a lateral meristem. And so it's fluid in the sense that, we have soft tissue here, but then as you go down, this is where the tissue in between, or the cells in between the vascular bundles, has begun to become meristematic, and it's made that meristematic ring, which then forms the lateral meristem, and then you have that increase in girth occurring. And so it's coming down, you have all of this has lateral meristem growth, and then of course this little bud took off and is growing, and it's all flexible growth. So this is just sort of the back to regular or initial orientation of that vascular tissue. You can see it occurs across the plant. You can go from having woody growth to non-woody growth up and down the particular plant. So I'm going to move in now and talk just a little bit about some of the different anatomical features of plants. So talk more about individual plant parts and some of the anatomy and some of the terminology with those plant parts, and also some of the functionality, what the functions of those different plants are. So first we'll talk about roots. All of these are pretty self-explanatory in terms of the functions of the root. We know, we talked earlier about some of their main functions, of course, are taking up water from the soil, but also minerals from the soil. There are some essential compounds that we'll talk a little bit later on about. And we also talked about they're often used for storing excess food. So those carbohydrates can often be stored in the roots for later growth and development. And then of course, they're the anchor. They're holding that plant often to the ground. So whether it's in a pot or if it's in soil, they're basically holding that plant secure. We already kind of looked inside the plant. We pointed out where the root apical meristem is, and what it's doing. It's undergoing mitosis, and it's making all the new cells. That's pushing that root further deeper into the soil. And then, of course, the orientation of that vascular tissue inside that root, whether it be a dicot in the center of the root, or if it's a monocot, it's forming more of a ring around the center of that root. Now we have some different types of roots in plants. We often refer to them either as a taproot, so a main central root that goes down into the soil, like a carrot. So a carrot is a great example of a taproot. Or we can see this more of a network of hairs called fibrous roots. So we often see in grasses, but many other plants. So those are often two very distinct types of roots. And we can see modified roots. So things that often don't necessarily look like a root can be roots. In this case, we've got a sweet potato, which is a root, a modified tuberous root. And it's got all of the anatomical features of a root that I just kind of showed you. So that's what makes it a modified root. If we move up to the stem tissue, or the above ground portions of the plant, of course, their main function is, it is storing some sugars in the stem tissue, but it's really supporting the leaves or the foliage of the plant, which is, of course, the manufacturers of where energy is being produced in the form of carbohydrates being produced. So it's supporting the leaves, which are undergoing photosynthesis, and then of course, it is conducting water and sugar throughout the plant. The other main parts of the above ground portions, we've used the word stem already. We've used the leaves. We've shown you the buds, the lateral buds or axillary buds, the terminal or apical meristem, and then a few other terms that we might want to also use now is node. And so an area on the stem where a leaf attaches to the stem, you have a node. That's just the region on the stem. That's what we call that. And where a leaf attaches to a stem, there's always a bud. And then the area between nodes is an internode. So we have a node here where we have leaves coming off of the stem. We go down the stem or the internode down to the next node where we have a leaf attached with a bud. And then, of course, as with modified roots, we can have modified stems. And so here's just two examples of some modified stem structures. Of course, everybody recognizes the top one as a potato. And so a potato or a tuber is a modified stem, and it has all of the stem parts, basically all of the same anatomical features that we just talked about. All of the eyes of the potato, those are all lateral buds. And then if we go down and look at a bulb, so in this case, we've got an onion, and an onion is a modified stem. So of course, we have its roots here at the bottom, and then we have this very compressed stem that has these modified leaves coming off either side. And so we just have this very compressed plant all enclosed in this little bulb. Now, modified stems can also take on a couple of other different shapes, and we can have ones that run across the surface of the soil, which are called stolons. So if you think of a strawberry plant, for example, it will send out little daughter plants out on these little runners, which we call stolons. So stolons are above ground lateral stems. And likewise, we can have underground lateral stems that send out runners to new plants, and those are called rhizomes. So in this case, we've got a parent plant here on the left. It sends out this little lateral underground stem called a rhizome, and then a new plant will pop up. So lots and lots of different examples of plants that have both of these. So of course, strawberries have stolons. Several different plants, including things like, if you've ever been to an aspen grove and you've seen all of those aspen trees, they're often from, a single individual or a few individuals that are sending out these rhizomes to new plants coming up. Okay, let's move on to talking about leaves. So, we've talked about leaves, and their, main function, of course, is to capture sunlight energy so they can undergo photosynthesis and make sugars for the plant, which are so critical for plants to be able to grow and develop. When we talk about their anatomy, a leaf is the whole entire structure, and that whole entire structure is often comprised of the big floppy thing, which is the blade, and then of course the little stalk that it sits on called the petiole. And then remember, where a leaf attaches to that stem, we have a little bud. Now there's many different types of leaves. We're not going to cover all of the different types, but I will cover I'll mention two main types that we often see in the plant world. The first is this category, what we call simple leaves. And so a simple leaf is basically a plant that has a single blade and a single petiole. So, and where it attaches to the stem, we have a bud. So of course, the little bud is going to be right down here, and we have a single blade and a petiole. Now we can also have what we call compound leaves. And so compound leaves are leaves that have multiple blades. So rather than just a single blade, it's going to have a grouping of blades. And there's two main types of compound leaves. One is called pinnately compound, and that's this example here in the middle. And the other is called palmately, so kind of like the palm of your hand. So these are also multiple blades. And the reason that, so this is each, in these compound examples, this is a single leaf right here in the middle. That's a leaf. And with the palmately compound, that's also a single leaf. What makes it a compound leaf is, again, the placement of a bud. So remember, I mentioned where a leaf attaches to the stem, there's a bud. So a leaf attaches to the stem. In the case of a simple leaf, there's a bud. Where in these compound examples, again, where a leaf attaches to the stem, there is a bud. So this whole thing, there's only one bud here, and it's right down where that whole thing attaches to the stem. The same goes true with this palmately compound. So it's the lack of buds where those smaller little blades all attach. So there's no buds in those up here in this pinnately compound leaf. There's no buds anywhere up there. And likewise, there's no buds where these little blades attach in this palmately compound. So that's what makes them compound and simple leaves. Now, another thing that I'd like to talk about when it comes to leaves is what we call their arrangement. And leaf arrangement, there's several different examples, and this is basically just the orientation or the pattern in which those leaves attach to the stem. And so in this first example, this is what we call alternate leaf arrangement, and that is where we have a single leaf attached to a node. So remember, a node is that area on the stem where leaves attach. And so if we just have one leaf attached at a node, we have one leaf, one bud at a node, and that is called alternate leaf arrangement. And you'll notice that as you go up an alternate leaf arrangement plant, you'll often see them kind of stair step up the plant. So you have one on the right, one on the left, one on the right, one on the left, and they'll basically alternate up the stem. Another type of leaf arrangement is called opposite. And so that is where you have two leaves attached at each node. So again, the node is where leaves attach to the stem. And if you have two, in this case, we have one on the left and one on the right, and we have two leaves. And there would also be two buds. So wherever a leaf attaches to the stem, there's a bud. So if we have two leaves, one on each side, there's going to be a bud on each side. So this is what we call opposite leaf arrangement. And then the last one is what we call whorled. And whorled is three or more leaves attached at a node. Likewise, like with the example that I just gave, if it has two leaves, and it's going to have two buds. And with a whorled arrangement, if it has three leaves or four leaves or five leaves, it would have the same corresponding number of buds. So let's go on to showing just a little bit more about the leaf and what's happening with the leaf and some of the different things that are occurring. So this is a microscopic view of a leaf from top to bottom. So this is the top, the outside of the leaf, and then we have sort of the epidermis. So this is that outer coating, the protective cells of the leaf. And then we have all of those cells that are undergoing photosynthesis, all of these cells that are stained kind of purplish red. And then you have As you go down, then you have these very distinct little kind of clusters of cells. And of course, these are the vascular tissue. These are the vascular bundles. So this is where the xylem and phloem is. And then going further down, then we get this very loose orientation of cells towards the bottom of the leaf. And at the very bottom of the leaf, again, we have the epidermis. But what's different on the bottom side is you can see these different cells that occur on the underside of the leaf that look a little bit different. And these are what we call stomates. And so stomates are very specialized cells that are important for regulating what comes in and what goes out of plant leaf. And so it's regulating carbon dioxide coming in, oxygen going out, and also water going out of the leaf through transpiration. And so that's one of the reasons why there's so much gap or empty space in the underside of the leaf here in between all these cells. It's a great place to store excess carbon dioxide to be used for photosynthesis as water's coming up through the vascular tissue to then be used in the chemical reaction of photosynthesis. So when it comes to these specialized cells called stomates, this is a picture of a corn leaf, and you can see these little puzzle-like pieces that all look very similar across the surface of the leaf. That's the epidermis, and so it's super tightly interwoven, so it's not letting anything in, letting anything out. But then you have these cells that look very different in the surface of the leaf called stomates, and these are what's allowing things to come and go inside the plant. And so they are highly specialized in that they will open to allow carbon dioxide in, and then they will shut. And then once photosynthesis occurs and it uses up all that carbon dioxide, it then has a bunch of oxygen that it just made as a result of photosynthesis. And then it basically opens, allows all that oxygen to release, and then allows more carbon dioxide to come in. So they're super important in the process of photosynthesis. And then they're also very important for regulating leaf temperature. So if the leaf or the plant is getting hot, they may open, allow water vapor to transpire to go out to help cool the immediate area right around the plant. So I'd like to move on to a different topic. And this is part of the first couple slides. I talked about this idea of plants being able to regenerate lost organs. And so I'm sure we've all done this or have seen this happen where we have our, maybe our favorite plant that we want to share with our friends or our neighbors. And so we take a piece of that plant and we take it off and we put it into a pot, and we come back, several months later, and then we have this occur, which we have basically this plant is starting to reform roots from this stem tissue. And so this idea of being able to basically regenerate lost organs is a process called adventitious growth. And plants can do this basically through a number of different ways, but the process is very similar in each of them, and it's all driven by these what we call internal regulators or plant hormones inside the plant. And so I'll show you an example. So if I was to take my Photinia, for example, a way that this could work is I could pluck this leaf off, and I can put it in a pot. And again, just like the last picture showed, I could pull this out after a couple months, and in theory, I could get little roots forming at the base of this. And so what happens is that at that injury site, a couple of different plant hormones will get rushed to that site of injury when it occurs, and it can tell the plant to de-differentiate those cells. So the cells that were initially there doing their thing, there was a petiole there that I just ripped off, and so there's all those different cells that I just showed you right here. So right here is some of those cells that are inside this leaf. So when that injury occurs, although these were all cells doing their thing, they were moving water and sugars, and they were undergoing photosynthesis, and they were protecting the plant. Well, these can then de-differentiate. So rather than being what they were just doing, they can go back into a meristematic state and undergo mitosis again, and basically cell division. And as they undergo cell division, they then form a new root apical meristem. And once that root apical meristem is in place, it undergoes mitosis. And then you have this whole process all pick up again from what I've described. And so it's a pretty remarkable process. It can happen from a leaf. Of course, it can happen just from, I can take a hardwood cutting, basically a chunk of stem, and put it into the soil and do the same thing. In agriculture and parts of agriculture, often they will take leaves and blend them up and make a juice, and they can put it on a little petri plate of growth auger and actually form a plant from that. So the process is all the same in terms of just having that initial parent plant material, a portion of it, to then get to start replicating again. So now let's move on to talking a little about another cool part of a plant, and that is the flowers. And I put this term inflorescence on here because we often just call the floral part of a plant the flowers, but you may often see this term used. And the inflorescence is the overall flowering structure, all of the flowers combined of a particular plant, and their pattern or orientation is what is referred to as the inflorescence. So whereas a individual plant may have what we call a single solitary flower that makes up its inflorescence, another plant may have a cluster of flowers that are in the shape of like an umbrella or an umbel inflorescence. So there's all sorts of terms that can be used to describe that shape or those types of flower arrangements. It's more important, probably used in plant identification. So if you're going to try to identify a particular plant, it may ask you to try to determine what the inflorescence looks like. So if we talk about flowers and what their goal is, of course, probably the main overarching goal when it comes to flowers is that the plant wants to undergo fertilization. It wants to have plant sex. It wants to undergo fertilization so it can form a fruit, form a seed, and carry on its genetic material to the next generation. And of course, plants and flowers do that through many different ways. In the case of some plants, it may require a flower to be pollinated by an insect. Sometimes it's wind pollinated. It could be bird pollinated. There's all sorts of different ways that pollination may occur. Some plants may have what are called the Nectar Guides to enhance the visitation by pollinators. So this bottom picture is an example of what a flower may look like to us. And then if you were to look at what it looks like to a particular insect, you can see that it looks very different. So it's very dark colors around the reproductive parts to try to attract that insect to flower. So we'll go ahead and look at some different flower parts here. So we'll start off, so this is an Alstromeria flower. And it's going to have the same flower parts that we're going to look at in the diagram here. So I'll just kind of show you both of them at the same time. So starting off at the base of the flower, usually most flowers will sit on some sort of a stalk that are called the pedicel. So in this case, you can see the pedicel or the stalk here on the Alstromeria. And going up the pedicel, we'll often get to a point that's fairly defined called the receptacle. And so in this case, it's this little swollen area that's still kind of greenish tissue. So that's the receptacle. And that's where pretty much all the other floral parts are going to come and attach. Now, if we start looking at the colorful parts of the flower, the first is what we call the sepals. And so they're often green in many flowers, and I'll show you an example of a green one here, just so you can see that. So this is a chrysanthemum. You can see this beautiful chrysanthemum here. And on the back side of the chrysanthemum, the sepals are really distinguishable here. So they're green. And this is what encloses the flower bud before it actually opens. And so if you think of a rosebud before a rose flower opens, you know, it's got the very distinct green sepals, and then as the flower opens, those sepals fall back. Same goes true for all these other ones. So in this case, in the Alstromeria, these are its actual sepals here. These actually have brightly colored sepals, so there's actually 3 of them. They alternate. There's 3 sepals with the Alstromeria. And then we move to the next whorl or the next group in, and those are the petals. So in the Alstromeria, it has 3 sepals and it has 3 petals. The sepals are just narrower than the petals. In other flowers, it's these that are the brightly colored things, whereas the sepals are green. It's the petals that are super bright. And then we get to the next whorl, which is the reproductive structure. So I'll take these petals off here and move them out of the way. So the next, again, all of these are starting to fall off, but I'll point them out here. We have all the reproductive parts that are left here. The ones that are all kind of starting to curl up here and fall off, these are the stamens. These are the male reproductive structures. So that's in the diagram here. It's these long filaments with these little yellow, to the anthers. This is where pollen originates, but these whole structures are called the stamens. So these are the male reproductive parts of the flower. So you can see the little anthers where the pollen is in the Alstromeria here, kind of off to the side here. So we have 6 of those, and I'll move those kind of off out of the way. So those are the males. And then we have the female, the pistil. So in this case, we have 1 pistil in the flower. So this is the female, and you can see it kind of wants to stick to my pen. This is the Alstromeria pistil here. And that again is the female portion of the flower. And there's a couple different parts of the pistil. The first is this little landing pad at the top called the stigma. So it can take on lots of different shapes. And so you can see in this one here with the Alstromeria, it's forked. So it has 3 little prongs. I think you can see those in the video. And then that is where the pollen lands. So in order for fertilization to even begin to happen, you need to get pollen from those anthers to land here on the stigma. And then just below the stigma is we have this long tube called the style. So this is the style of that's the middle portion of the female. And then at the very base of the pistil is the ovary. And so inside the ovary is, of course, where the ovules are, and that is where fertilization would occur. So in this case, I'll cut open the ovary here, or if we just zoom in just a little bit here. So hopefully you can see. So inside the ovary here, you can see all these little dots. So those are all the ovules inside this pistil. If we, again, go back to the whole female, I'm going to show you a video of it. But so pollen, in order for fertilization to happen, pollen's going to have to land here on this little landing pad, the stigma, and then it's going to grow basically a tube. And this pollen grain is going to grow a tube down the middle of the style, around the base of the ovary, down to the bottom here. And then once this tube is in place, the little male gamete in the pollen grain is going to get transferred down that tube up into where the ovules are, and then hopefully fertilization will occur. And that has to happen for every single one of these little ovules. So lots and lots of little pollen grains are going to land there, and lots of them will grow these little tubes all the way down there in order to then transfer the gamete. Okay, so as I showed you in that last example, both in the diagram as well as the Alstromeria, both of those flowers had both male and female reproductive parts. And so what we call that is a perfect flower. So if a flower has both reproductive parts, it is a perfect flower. Now, we can also have a plant that does not have both reproductive structures, and that is an imperfect flower. So in the picture here on the left, we have, of course, very similar to the flower that we dissected, but also the flower that was in the diagram, and that was an example where it had both the male and female reproductive structures. And then again, on the right here is what we call an imperfect flower, where it's only going to have one sex. So it's either going to have the male parts, which we would call a staminate flower, or it will have the female part. So it would just have a female, and that would be called a pistillate flower. Now we can take this idea of sexes in flowers to the sex of the plant. The first term that we're going to talk about is called monoecious. So monoecious basically means one house. So one house plants are plants that have both the male and female reproductive structures on the same plant. So it can undergo fertilization on that plant, produce a fruit with seeds in it. Now, if you think about a monoecious plant, you can get to that two different ways. You can have plants that have both reproductive structures in the same flower. So they can have a plant that's made up of all perfect flowers that have both male and female parts in all those flowers. So that would make it monoecious. But you can also have a monoecious plant that has imperfect flowers, both male and female imperfect flowers on the same plant. So if you think of something like a melon, squash, cucumbers, anything in that cucurbit family of plants, you'll have a big plant with lots and lots of flowers on it, and some of those flowers will be females and some of them will be males, but they all occur on the same plant, so that makes them still monoecious. Now, the opposite of that is what we call dioecious plants. So di means two, ecious means house. So these are the two house plants. And so that's where we actually have separate male plants, separate female plants. So you're going to have some plants that just have stamen flowers or staminate flowers. Those are the male reproductive flowers. And then you're going to have completely separate plants that are female flowers that just have those pistillate flowers. So some examples of those would be, if anybody has ever had like a ginkgo tree, you'll have male ginkgo trees, female ginkgo trees. Kiwi is another, if you've ever grown kiwi or have seen kiwi plants, you'll have male plants and female plants. And same with asparagus, you'll have male and females. So this is really important when it comes to gardening, because if you're relying on, if it's a plant that produces fruits, like in the case of a kiwi, you'll need female plants, right, to be able to produce fruits. But in order for those fruits to even get a chance at, undergoing reproduction and then forming a fruit, you need to have some male plants. So as I mentioned, the ultimate goal, of course, with flowers is fertilization. And I started to talk about the process of what happens with pollen from the anther being deposited on the stigma, and then that pollen grain growing down that tube, the style, down to the base of the ovary. And then once that tube is in place, the male gamete can then be moved down that tube to where the female gametes are to undergo fertilization. And so here's just a little graphical depiction of that, showing how that pollen grain germinates and forms this little tube, and then transferring that gamete down to the base of the ovary where fertilization can occur. And then, of course, once fertilization occurs, we get the formation of a fruit. And inside that fruit, we get seeds, and the seeds are that product of sexual reproduction. And inside that seed, of course, is the genetic material from both plants, or in the case of both flowers. And it's going to have basically this little embryonic plant in there that's going to then the start of the next generation. So we used the term dicotyledonous plants and monocotyledonous plants earlier on today. The cotyledon, that is what we call the seed leaf. So inside that seed, there is a little seed leaf. That's basically you could think of it as the gas tank, right? It's got basically this little gas tank of carbohydrates in there that's going to be the supply of carbohydrates that get this plant initially growing until it can start making its own true leaves. And so there'll be some seeds that have one cotyledon, and those are the monocots, or there'll be plants that have two seed leaves or two cotyledons in their seeds, and those will be, of course, the dicotyledonous plants, and that's where that term comes from. So, of course, we have lots and lots of different flowers, and they can take on many, many different shapes. And as we start to look at those flowers, they will begin to tell us, a bit more about that plant. So let's go ahead and take a look at a sunflower for a moment. So I'd like to show a picture of a couple of different flowers and get a sense of just how much diversity there is in plant flowers and plant parts. And so this, of course, the picture here is a sunflower. And I have here on the video an example of a plant that's also in the sunflower family. And what looks to be just a single flower for many of these members of the sunflower family are actually something much more. And so if we start looking at plant parts and actually dissecting these, we would find that if I pull one of these off here, oops, let me go ahead and pull that off. So if we look at the members of these sunflowers, we would find that this here is actually a whole complete flower. So that's one flower. And how I would know that is if I look down in here, there's a reproductive part down there. So in the case of these members of the sunflower, these are often imperfect flowers. So this is an imperfect flower, and this is a staminate flower. So these ring of flowers that occur around the perimeter that we often think of, these brightly colored petals here, these are imperfect flowers. Often they're staminate flowers, male flowers. And then these are called what we call ray flowers. That's the kind of flower they are. And then if we look closer inside here in the center, all of these, okay, all of that, all of these individual things, these are also flowers. All of these are individual flowers. And in each one of those is a reproductive structure. In these, it's the pistil. So these are imperfect female flowers inside the center of these sunflowers. So if you look across this sunflower here in this picture, all of these are called disc flowers, and all of those are pistillate flowers. So if you think about a sunflower, if you've ever seen them produce seed, the seeds all occur across the middle here in these disc flowers, because these are where the female flowers are. And then the pollen, of course, is coming from these male flowers out here where these big petals are. So there's just a couple more pictures showing those close up here. So again, we have those male flowers out here, and then all of these are individual flowers being disc flowers in there. You can see we have just tiny, tiny petals that are all fused together to form a little tube with the little female pistil being in the center. Okay, so finishing up this little section here now, part of fertilization, of course, is the end formation of a fruit. So once we get fertilization occurring, the fruit is the plant part that contains the mature swollen ovary and its seeds. So we showed you what the ovary part of the the flower is. So the fruit is often derived from that ovary and parts of surrounding that ovary tissue, and then inside of that being a seed. And there's all sorts of different shapes and sizes of fruits. And many of you have probably heard of the different types of fruits that are categorized in plants. You may have heard of the word pome before. That's a type of fruit that we see that come off of plants like apples and pears and pomegranates. Drupe is another type of fruit. That includes many of the things that have stones in them, like a peach or almonds or apricots or cherries or olives. There's multiple fruits. There's nuts, of course. Nuts is a big classification of fruits. And then we have berries, which are things like cranberries, grapes, and kiwis, and also aggregate fruits, which are all of the brambles. So like blackberries and strawberries are all types of aggregate fruits. So here's just a couple, two different examples of those classifications of fruits and some of the different terminology that's used to help describe these different classifications. And so, a fun example, of course, the picture on the left is an example of citrus, and citrus are actually berries, a classification of berries. So, whether it be lemons or oranges, they are all berries. And the picture on the right is an example of an aggregate fruit. So things like strawberries, and in this case, raspberries, these are all aggregates. And of course, aggregates form by sugars being filled inside the middle of the flower structure. Remember the receptacle, that part of the flower part where all of the other parts come, like this, the sepals and the petals, everything came and attached to that receptacle part of the plant. That's what fills up with sugars in the case of these aggregate fruits, and then it pushes everything out to the surface. And so if we look at that, this is an example of a strawberry flower. And if you look at a strawberry flower, of course, we know all of these parts. We know the sepals back here, we know the petals, and then we have this ring of stamens that go around the outer whorl. And then inside here, all of these little yellow things, these are all pistils. So this flower has lots and lots of stamens, lots and lots of pistils. And as this fruit, or as this plant undergoes fertilization, and then we start forming the fruit, remember, this is an aggregate fruit, meaning that the receptacle where all of these things attach. So if you think of the backside of this strawberry flower, where all these plant parts attach, there's that receptacle. And so it's the receptacle that starts getting filled with sugars, and it just keeps getting filled and filled, plump full of sugars, and it basically pushes everything that's attached to it out to the outer surface. And that's what we get when it comes to a strawberry. So when we look at a strawberry here, of course, we know the sepals and the petals, the receptacle was right down, the base of the receptacle was there. And as you can envision it getting filled like a balloon full of sugars, it pushes everything that was attached to it to the surface. So these are all these little brown things. These are all pistils. These are all the female pistils that were all right here. And they just kind of all got pushed out to the surface of this fruit. And so it's actually the inside of these little brown things where you actually find the seeds. So these aren't just the seeds themselves. The seeds are actually inside these little dry structures called achenes. And so this is the same whether we are looking at a strawberry, or in the case of a blackberry or a raspberry, the process is exactly the same, but rather than them being dry, so rather than these being dry like in a strawberry, they are fleshy when it comes to a raspberry or a blackberry. So again, the receptacle gets filled with sugars, it pushes all of those pistils to the outer edge, and it's inside those little drupelets out here, these little juicy, in the case of raspberries, they're all juicy, and inside that is the seed. So that's why we don't see seeds like on the inside of a strawberry or on the inside of a blackberry or raspberry, because how the fruit forms, it pushes all of those to the surface of the fruit. And that's where you'd see, of course, the seeds. Like in a blackberry, it's in those little juicy pods out on the very outside of the fruit. Okay, so let's talk a little bit about fruit formation is dependent on the parent plant. And so I've got an example here where I've got a gala apple tree in my yard, and my neighbor also has a gala apple tree in their yard. And often we get cross-pollination, right? So gala pollen from their flowers comes and lands on my gala tree and vice versa. But when I pick the fruit off of my tree, of course, what do I get? I get a gala apple. Now that all, of course, all makes sense. That's very logical. But what if my neighbor has a Fuji apple tree and that Fuji pollen lands on my gala gala flowers and I pick the fruit? Well, I still get a gala apple tree. I still get a gala fruit from my apple tree. And that's because the fruit is completely dependent on the parent plant. So its shape, its texture, its flavors, all of those things are dependent on the parent plant. However, it's the seeds that are inside that fruit. That is where you would see characteristics from both the source of the pollen and also the parent plant. So if we were to grow the seeds from these two different fruits, we'd get trees that would have fruits that look very, very different. So that is where we would see the difference in the genetic variation. So I'd like to transition now and start talking about those plant hormones that we mentioned earlier on. And so these are internal chemicals that are formed inside the plant. They move around the plant and they regulate different parts of plant growth and development. And there's 5 different classes of plant hormones, and we'll talk about each one of them individually. The first one that we're going to talk about is called auxin. And so auxin is a hormone that originates in the tip of the plant. So this is in remember the shoot apical meristem, the terminal bud of the plant. That's where auxin is produced. And then it moves down the plant basically by gravity, by diffusion. And so at the tip of a plant, we have lots of auxin. As we make our way down the plant, we get less auxin. And some of the things that auxin is responsible for is it directs shoots towards light. So if you've ever seen a plant in a window and it's leaning, towards that light source, auxin plays a part of that. It also is responsible for what we call apical dominance, or suppressing lateral branches. It also is involved with preventing fruit drop. And then it is the chemical that is really important for inducing those adventitious roots. So remember, we take a leaf, we stick it in, some rooting media and try to get it to root. Auxin is really important in that process. And then the last thing that's kind of interesting is that at high concentrations, it's actually a broadleaf herbicide, so it can actually kill plants. So here is a video showing auxin at work. So auxin stimulates cell elongation. So it's being produced at the tip. And if that tip is laying on its side, it's going to concentrate more on the underside of this stem, causing those cells to elongate faster, causing that plant to bend upwards towards the light. And so that is one cause of auxin. The other that I mentioned is apical dominance. And so I mentioned auxin will suppress lateral buds or lateral branches. So auxin is produced here at the shoot apical meristem. And so high concentration of auxin up here. And so there's a lot of suppression of lateral buds below that auxin source. So of course, these branches are going to be short because there is a lot of influence of auxin up here. And then as you move your way down the trunk, you get less auxin because it's further away from the source. And those branches are going to be bigger because they are not being suppressed as much. But what happens if we remove this terminal bud? We remove the auxin source. Usually what you'll see is this rapid growth of the buds just below it because it's removed that suppression. And so we see this when we're pruning plants, right? So I've got a plant here but I'll show you. Here's a plant that I pruned a few months ago. So I came in here, pruned this plant, and just in the last month, what has happened? These lateral buds, so there was a bud there and a bud there, they have now been released, right? So I removed the auxin source. So that's typically what you see when you go in and do winter pruning, right? You prune, and then all of a sudden when growth starts, you just get this flush of growth coming out of just below or even at that cut end. And that's, again, because the auxin source was removed. So the other time we see this often important is in, with plants where we want to make them bushy. So if you have a plant like a coleus or other plants where they get a little leggy, you can go in and you can tip them. So you tip by taking off the tips of all of the terminal branches. And then of course it allows those branches or the buds further down on those branches to begin to grow and express themselves. So this is extremely important in pruning, and you guys you'll probably see more classes specifically on pruning and how removing the terminal leaders and removing the tips of the apical meristems can be really important with forming good structure when it comes to trees. Now, I also mentioned at low concentrations, auxin can be important for stimulating adventitious growth. And so up here in the right, this is an example of root tone. This is a rooting hormone. And so this is either NAA or IBA. These are two formulations of an auxin product that basically mimics or is the same as auxin in plants. And so by dipping a plant in root tone, it'll help stimulate that root formation. Likewise, I mentioned at high concentrations, it can be a broadleaf herbicide. So things like 2,4-D or MCPA, these are auxin mimics. And so at high concentrations, basically it tells the plant to grow, elongate. And so often you see these broadleaf plants twisting and curling and cupping and basically growing themselves to death. The second type of plant hormone that we'd like to talk about is gibberellins. And so gibberellins is a shoot and fruit stimulator. So it's used in agriculture to make giant plants. And so we use it much like we use auxin. We use it synthetically as well to help make fruits bigger. Naturally, it is used to kind of regulate shoot and fruit growth, but it's also used for seed germination for seeds that are pretty hard to germinate. So it may have a hard dormancy to break. You can use gibberellins to help break that dormancy. So the picture here is, of course, Thompson seedless grapes. And if you've ever tried to grow Thompson seedless grapes, you often get these very tight clusters with little tiny berries. And it's often because you're not applying gibberellic acid. So in agriculture, one of the ways that Thompson seedless grapes are made to be really big, loose clusters with these huge, massive berries is the plants are treated with gibberellic acid during the early stages of fruit development. Now, the third plant hormone that I'm going to cover is what we call cytokinins. So cytokinins is one of those plant hormones that originates in the roots and then moves upward. So unlike auxin that is made in the tip of the plant and moves downward, cytokinins is just the opposite. It's made down in the roots and then moves upwards. And some of its major functions is that it helps govern cell division. Also, it's helpful in that regeneration process and then correlating root and shoot growth. And then we have ethylene. So ethylene is the only plant hormone that's a gas. So as those other ones are in the liquid phase moving around the plant, ethylene is actually inside the plant moving around as a gas. And I would say the primary function of ethylene in plants is that it's a ripening hormone. And it's also really important as an injury signal. So when a plant is injured, that's one of the hormones that rushes to that site of injury to help repair and defend against, basically walling off that site of injury to basically keep the plant from allowing bad pathogens from entering the plant. And so it's used in, of course, agriculture for lots and lots of different reasons, mostly for the ripening. So, if you ever buy bananas, they're often gassed with ethylene. So they're picked super green, gassed with ethylene to promote that ripening process to make them ripen. And then we have the last one here, which is abscisic acid. And so abscisic acid is important as a stress signal. Also, when we think about plants that are getting ready to go deciduous, so plants that are going to lose their leaves during the fall and winter months, there's actually a part on the plant itself called the Abscission Zone, where the plant will naturally want to wall off or drop that particular organ, in this case, a leaf. So when leaves are getting ready to fall, the Abscission Zone walls that leaf or that organ off, causing it to drop. And it's extremely important for a stress signal for plants that are stressed. So we talked about stomates, and abscisic acid is really important for causing those stomates to close in response to stress. So here is a picture of the Abscission Zone. So this is just to show where, like a leaf, in this case, a leaf is attaching to the stem. There's the little axillary or lateral bud. And then it's these specialized cells here that make up the Abscission Zone. So when it's time for the plant to drop its leaves in the fall, it's abscisic acid that causes these cells basically to die off and form this basically protective layer that will then shed that leaf. And then we often see what the result of that is, of course, a scar, a leaf scar. And that's the Abscission Zone. Okay, I'd like to finish up today with just talking a little bit about some external responses that affect growth and development. And so where we just talked about internal regulators being the plant hormones, these external regulators are often referred to as tropisms. And these tropisms will cause plants to either grow differently or alter their growth to basically capture more energy, capture more water, uptake more nutrients, many other different examples. And so the first tropism that I'm going to cover is called phototropism, and that is where the external source is light. And so earlier I talked about auxin and how auxin can bend a stem upward. And so with plants that have phototropism, you'll see in the picture here, so auxin is made in the shoot tip and moves downward by diffusion. And if a plant is growing upright, those auxin molecules are fairly even going down the stem. With plants that have solar tracking, the auxin actually accumulates on either side of the stem, causing it to bend, having those cells elongate more, causing the plant to then bend towards the sun. And there's lots of examples of plants that show phototropism. That includes, things in the sunflower family have a tendency to be solar trackers, also eucalyptus trees, and some members of the legume family, especially lupins, are really good at solar tracking. Now, the second tropism is what it called gravitropism. And so gravitropism, of course, is in response to gravity. And this is most apparent, of course, in roots, where we know roots grow down. And there's these little structures in roots called otoliths. And those otoliths, of course, will settle when a plant is growing, the root is growing horizontal, the otoliths will settle on the bottom. And in response to that, the actual root will grow and bend down following gravity. And then the last one here, we have thigmotropism. And so thigmotropism is in response to touch or some sort of a mechanical signal like pressure. And there's a few different plants that come to mind that have great examples of thigmotropism. The first one is the plant here on the left. This is, of course, the touch plant. And when this plant has some sort of either touch, when you touch the leaf, or if a strong breeze or wind hits the plant, it causes this immediate change in the turgor pressure of the plant, causing the leaf to completely collapse. And then the other plant here is, of course, the Venus flytrap, which is here on the right. And there are, of course, some specialized triggers in these modified leaves. So when an insect passes over these triggers, it'll cause the leaf to very quickly close, capturing that meal. So another great example of thigmotropism. So I'd like to finish off here just thanking you for listening to this lecture. And there's some great resources on for introduction to horticulture, including the California Master Gardener Handbook. And then also there's several other resources out there, including Botany for gardeners, as well as your local cooperative extension system. Thank you so much and take care.